Okay, I know y'all just ate. So I've only got half the number of slides that we had on the other two PowerPoints. So we're going to get through this really quickly. Is that all right? So we can make all of this work. I just want to talk a little bit about evolution and the science for creation. Um, Not the science for evolution. I'm going to talk about the science for creation. And so we'll just look at a few things there. When I first started teaching evolution, it was 1995. I didn't believe in evolution at all, and I didn't know really how to teach the course. And at LCU, we used to call it evolution creation, but then that would not transfer to any other university. So we had to just call it evolution. And of course, our students need to know evolution because when they take tests to get into med school or dental school, they have to know evolution. So we have to teach the evolution. So I didn't know how to teach it. But there's this one guy, and you can see his book up here. His book was Evolution, a Theory in Crisis. And Michael Denton is a neurologist down in Australia. And he just found all kinds of problems with evolution. And he wrote a book about it. So that was my main textbook that I used for this course. And we went through it, and that's how we got through the course and figured out how to teach it. But he wrote another book in 2016. It's called Evolution, Still a Theory in Crisis. And so things have not changed. Evolution is not coming up with any data. In fact, ever since 2012, that world has been silent. And I'll tell you why here in just a minute on all of that. And so then came along Michael Behe, his book, Darwin's Black Box. It came out in 96, and then he revised it again in 2006. Just a great guy. He was not a creationist. He was an evolutionist, and he was doing work on the bacteria flagella. So the flagella is that thing that whips around that propels bacteria through the water. And he was looking, he's a microbiologist, and he was looking at this bacteria, and he got to see how that flagella was working and all the stuff involved. And he said, I wonder how that came about by evolution. And he was just curious. So he checked the literature to find out how did this motor, and y'all can kind of see it up there, this motor come about that turns around, that propels that bacteria. And he searched everywhere in every evolution journal he could find. He found nothing. He found nothing. And he said, that thing, it has a rotor, it's got a stator, it's got everything that you would find in a motorboat motor. This looks like something that was designed. And so he wrote the book, Darwin's Black Box. And he said, he looked at this, he looked at other things, and he said, this had to be designed. Turn the biology world upside down. Here was a guy who everybody thought was an evolutionist, but he said, evolution can't explain all of this. And so I was really appreciated. This particular motor has over 40 different proteins, and it just works like a motor. And so I'll show you some more of that tomorrow when we look at that. But Behe calls them irreducibly complex machines. That's what he calls them. And what that is it's a machine composed of several parts, and all the parts are working together and had to be together from the beginning to work. And if you take away any one part, it doesn't work. That's an irreducibly complex machine. And he says, we see that everywhere. And he even went through how we clot blood. Whenever we get a cut on our hands, whenever we get a cut, it will cause that factor, it'll cause that factor to be converted to that one, and that causes that one to be converted to that one, and that one to be converted to that one. It's just a cascade all the way down to clotting blood. There's about 20 different proteins that are necessary, and they must follow an order. And if you take out any one of those factors, you are a hemophiliac. It doesn't work. That's an irreducibly complex machine. And so that's what he found in lots of biology examples. So he wrote his book, and he's still going strong. I'm going to tell you a little bit about his third book here in just a second that we're talking about. First, let me switch to evolution's story. Here is their story, and I'm just going to spend a little bit of time here on this. Evolution's story is this. You can break it down to three parts. There are mutations. Some are good, some are bad. Natural selection occurs. We know there's competition in nature and certain mutations will better fit the environment. So we know natural selection occurs. And then selection, if you repeat it over and over again, it's going to lead to a new species. And in that process, build up all the wonderful structures of life. So that's evolution's story, how we went from a bacteria to all the complex things we had today. There were mutations, there was natural selection working on that. And if you just repeat that natural selection over and over and over, and you have mutations coming in and better mutations that better fit the environment, you can come up with a new species and then another new species and another new species. That's evolution's story. Well, there are mutations. That part is true. There are mutations. They say some are bad, some are good. Well, it's not what you think. Laura, you can answer this. For every one beneficial mutation, how many harmful ones do you think there are? You can't answer that either. You already know that answer too. Anybody want to guess? For every one good mutation, how many bad ones are there? 1,000? Anybody else want to guess? 500 million. You're really close. It's 1 million. It's 1 million. So there are mutations, but to get a good mutation, 1 out of 1 million. And then it's not what you think even after that. And I'll show you that here in just a minute. Natural selection. Yep, it occurs. We know natural selection occurs, but it's very limited to within a certain genus. It's just within a certain genus. It doesn't work at the family level or any level up above that. And so that'll become important in a minute, and I'll talk to you about that in a minute. So evolution is using two things that do happen. Mutations do happen, natural selection happens, but that third point, it's never been shown. It's never been shown. We've never seen that in biology at all. That is what they stick on to match what they want it to be. So there are mutations in natural selection, but it kind of works opposite of what they want it to work. And that's what I'm about to show you, okay? So we're going to look at several organisms. We're going to try to understand natural selection and the genetics behind all of this. The polar bear. Anybody like the polar bear? Yeah, they're kind of fun. Lives up in the Arctic region where it's really, really cold. It's really closely related to the brown bear. In fact, they were the same species, genus species, for a long time up until the 1700s, and then they were separated and made two different species. They can still interbreed with each other and produce viable offspring. So they're really the same species, but they call them two different species because they look so different and because they live in two different environments. And I'm okay with that. I'm okay with that, calling them two different species. Now then, so if you understand biology, you kind of got to understand in the animal kingdom, we break up the animal kingdom into different phyla into different classes. I'm just really not good at this at all. I figured I could get better, but I'm not. And we divide up classes into orders and orders into families, and then finally families into genus and species. So there's the bear family, and there's the brown bear, and there's the white bear. Everybody follow that kind of? Yeah, that's how they classify organisms. And so they're in the same family, the bear family. Supposedly, the polar bear branched off from the brown bear, got lighter in color, more resistant to cold, and more adapted to be able to eat seal blubber up in the Arctic region. Every character here offers an advantage over the previous steps of evolution in making the polar bear to fit that Arctic environment. So, this is kind of fun. They did the genetics. Y'all know now we can sequence anything. We've sequenced our genome. We've sequenced almost every genome we know of. So we can sequence genomes really well and then look at the genetics. What is actually different in the DNA of the polar bear and the brown bear? What is different? It's mainly two genes. One gene was the APOB gene. It made the bear more likely to eat fat. So it actually changed the LDLs, that's part of your cholesterol. It actually changed the LDLs to where they're not carrying cholesterol, so you can't get heart attacks. And so it can eat all the seal blubber it wants to. And then the other gene was LYST, and it's associated with coat color. Changes in that gene are responsible for the loss of color in the fur. Both of those genes broke. And that's what gives us the polar bear. So the brown bear and the polar bear have the same genes, they have the same genetics. It's just two genes broke, and that gave us a polar bear. Isn't that interesting? Evolutionists want us to think that we've added on new genetic material. We've never seen that. We've never seen that in any organism. The reason we're getting beneficial mutations is because genes break. The genes are there all the time, they just break. Is that not interesting? Yeah. So the polar bear adapted to that harsh environment by breaking a gene that its ancestor had. So evolution doesn't work by adding new genetic material, rather by broken genes. But the interesting thing is, once that gene broke, now that polar bear is limited to the Arctic region. It can't evolve very much more because it's limited to that region. It can maybe break a few more genes and change a few traits, but it's pretty much limited to the Arctic region. Does that make sense? That's how evolution works. Every example they give in evolution, it's broken genes. Isn't that interesting? Yeah. So sequencing the genome has been a blessing in this particular world because we get to see all those things. We see the same thing in humans. Sickle cell gene, that's just a single base change in the DNA. But if you live in Africa, you're resistant to malaria if you have that. So that's a beneficial mutation, but it's a broken gene. For us to have that broken gene, it's not very beneficial because we can't carry oxygen very well on our red blood cells because the red blood cells are sickle shaped. But for people in Africa, beneficial mutation. Good mutation, but it's a broken gene. The OCA2 gene, when that gene breaks, it doesn't allow melanin to be produced in your eyes. You have blue eyes. So for a lot of people, having blue eyes, that's a good thing, beneficial mutation, but it's a broken gene. Y'all getting my point? So now with our new molecular techniques, we can look at the genetics of what's going on in all of these organisms that they say have evolved. So we can test Darwin's mechanism of mutation and natural selection. But what we're finding, it's always a broken gene that gives a beneficial mutation. That's really kind of fun. So that means if you go to Genesis 2, not Genesis 1, but Genesis 2, thus the heavens and the earth were completed in all their host. God, by the seventh day, God completed his work which he had done, and he rested on the seventh day from all his work that he had done. Then God blessed the seventh day and sanctified it, because in it he rested from all the work which he had created and made. All the DNA that has ever been made was made during the creation week. Have things evolved? In some cases, like the polar bear from the brown bear, but it didn't add on any new DNA. It just broke a couple of genes. Is that not interesting? Yeah. So once we figured all this out, which is around 2012, the evolution world has been silent. They don't know what to do. They don't know what to do, which is really fun, just really fun. So Darwinian evolution proceeds mainly by damaging or breaking genes. Darwinian processes can only account for small-scale adaptation, the polar bear to the Arctic region. That's all it can account for, not changes above the family level. So, just so you will know, when we talk about the family level, y'all know we've got the dog family, we've got the cat family, we've got the bear family, all these families. That is real similar to kinds that we read about in the Bible. Those are the kinds. And so, yeah, you can have different genuses in that family. We have the polar bear, we have the brown bear, we have the koala bear. They're all in that family. So that was the kind taken on the ark. And then since then, we may add a genus or two along the way, but it's always a broken gene. Does that make sense? So is evolution occurring? Yeah. The evolution of the polar bear from the brown bear, that does look like an evolution thing, but it's a broken gene. And now that polar bear is very limited to where it can live. So it's worse off than before as far as being able to live in a lot of different situations. Is this making sense? Yeah. So it's just really kind of fun. You can read about this in Michael Behe's third book, Darwin Devolves. So now that we're finding out how evolution works, and he goes through several examples, and I'm going to show you here in a minute, all the examples of evolution that they use, it's always a broken gene. It's just a broken gene. The Darwin's finches, they're on the Galapagos Islands there. There's about 12 to 14 different species of finches. That's what they look like. They differ in beak size and color, and some are lighter in color, some have longer beaks, some shorter beaks, some pointed beaks. There's differences among those finches. Well, let me show you where they're at. Do y'all know where the Galapagos Islands are? They're right there. So Darwin left England, came down here. I'm not good at this, I'm sorry. He came around here on South America, but when he got to the Galapagos Islands there, he noticed on those islands that the finches on those islands were really similar to the finches on the mainland. They just got blown over somehow to the Galapagos Islands and changed. And that's what Darwin was thinking. And I'm thinking that too. These finches just got blown over to the islands. There's a lot of different ecological niches there. They just changed a little bit to fit those niches. That's evolution. So yeah, evolution was occurring there. But how did that occur? Well, they've done the genetics on this. This is what they show in the textbooks. This is what is in our textbook today. They looked at the finches and their beak size. You can see they measured the beak size. And in 1986, the average beak size was about 8.8 millimeters from the top of the beak to the bottom of the beak. And then the offspring in 1986 was that same 8.8 millimeters. But then they had a drought. And in 1988, I'm reading the right years, aren't I? 1978, it was 76. And then 2 years later, after a drought, the beak size increased a whole millimeter to 9.8 millimeters. So the technical definition of evolution is a change in a certain character in a population. And so this character, beak size, changed for the whole population. What happened is almost all the birds died. When it was a drought, the small plants could not live, but the large plants that had deep roots could get down to water and they survived. So there were a lot of large seeds, but there were no small seeds. So all the birds that had little beaks died, which meant most of the population now has large beaks. So beak size went up. Does that make sense? Technical definition of evolution, a change in a character over time in a species. So it evolved. So it did evolve. It got larger beaks. And that's what they show our kids in the textbooks. Well, I have a good baloney detector. I know to look up the data. So I went and looked up the data of these finches that the Grants researched. And there's the data. And sure enough, look at this. In 76 to 78, beak size went up, but then it rained. It came back down. Then it went up, then it went back down, stayed down a long time, stayed down a long time, went back up. Beak size just goes up and down in any population. It's because the environment changes. So in reality, beak size never really changed over time. Yeah, we have some that have small beaks. We have some that have large beaks. Look at humans. We have some that have big heads. I've got a big head. We have tall people. We got short people. We just have variation. And sometimes the population moves this way. Sometimes the population moves this way. But in the world of evolution, they're saying it's evolving. They're giving us the sense that it's changing. They're giving us a sense that these finches, once they got blown over to the Galapagos Islands, They changed and evolved. And then they can get in another situation and evolve again, and then evolve again and become something else. But in reality, what is happening is they evolved, a few genes broke, and let me just show you. They kept the DNA of all these birds through time, and they figured out there were 6 master genes for head and beak development. One was ALXI, and 2 mutations in that gene was the difference between pointed beaks and blunt beaks. And they figured out that it was broken genes. They figured out in all of those genes, it was only the broken genes that caused a new trait. So what happens here is these finches get blown over from the mainland to these islands. They change because it's a different environment. And so some genes break, and that gene breaking makes this finch fit this particular ecosystem. And there's a lot of little niches on islands. And if another gene breaks, it makes this bird fit this ecosystem a little bit. But now those birds are limited to just that area. For them to change much more, they probably can't. But Darwin was thinking, whoa, they changed. Maybe they can change some more and change some more and change into another organism. Do you see the difference? We're all looking at the same thing here, but he's thinking that way, but he didn't know the genetics. Now that we know the genetics, every example of evolution they show is a broken gene. Isn't that cool? God made all the DNA at the very beginning. Nothing's changing. Yeah, some genes break. That just happens. May give a new trait. So you might say we evolve a little bit, but then we're very limited once that happens. So it's just been fun being in biology here recently. This has just been really fun. The cichlids, the fish in Africa, in the lakes over there, they say this is one of the greatest evolution stories. It's evolved, and now we've got like 400 different species of cichlid fish. Well, they did some genetics of that. One mutant gene, EDNRBI, was reported. It mutated in 2 regions, and the 2 traits controlled by the normal protein were lost by the mutant. They broke. The genes just broke. But it gave a different looking, there you can see they're all different kinds of different looking cichlids, but they came about because of broken genes. Isn't that interesting? It's just fun. This is fun. I talked to another guy who really worked in cichlids, and he said most of the changes can be identified on 4 different genes. And you can get multiple mutations in one gene that'll affect several different traits. And it'll give a new looking fish. It's still a cichlid, but it's a broken gene. And it's not going to change into any other organism. It's just going to stay a cichlid fish with a different trait. Does that make sense, everybody? Yeah, so it's just fun. This guy, he's a professor at Michigan State. He decided he was going to look at mutation in bacteria, single-cell bacteria. So he got 12 flasks, lined them up, and every day, y'all know this is E. coli bacteria. It's in our gut, and it divides every 20 minutes. So you can start out at the beginning of a day with 1 bacteria, and by the end of the day, you're going to have millions of these bacteria. So he started out with the same bacteria in all 12 flasks. Those are flasks that you can see up there. Every day, he'd take out 1% of the bacteria and put it in a new flask. And he just does that every day. And he's been doing this for 25 years. And that equals 65,000 generations. For an organism like a human, we're talking millions of years. So there should see some kind of evolution. That's what he was wanting to see. We were wanting to see some kind of evolution. And they did. They started growing faster. After 50,000 generations, they were growing 70% faster than at the beginning. And they got fatter. There was an 85% increase in volume. So these bacteria were growing faster and getting fatter. They were changing. They were evolving over time. And after they did the DNA analysis, they looked at all the DNA analysis. All the mutations they tracked were broken genes. Isn't that fun? This is just fun. They looked at 59 genes. Most of them changed mostly by degrading. So it's just, that's how evolution works. A few genes break, but then that limits that particular organism to just that environment that it can live in. So it's just broken genes. There's other examples. Horses that trot, I don't know if anybody has horses in here. The smooth trotting horses is due to a broken gene. A third of that gene broke off, a gene in the nervous system, and that makes the horse run smoother. So that's a beneficial trait, but it's a broken gene that caused that. The woolly mammoth, this just came out, and I just think this is really fun. Yes, it's connected to all the other elephants, the Asian elephant, the African elephant, all of those, but it's got 87 genes that broke. 87 genes that broke, and that gives it its look that it's got. It was genes that dealt with skeletal morphology and with hair growth. And you can see up there, it shows that the yellow gene up there, the DROSA gene, it just broke and just wasn't there. It wasn't working. So now we know the woolly mammoth is just an elephant, but it's got 87 broken genes. No new DNA, just broken genes is all it is. And that limits that woolly mammoth to a certain ecosystem. That's the only place it can live. The New York Times, in humans, some people are immune to developing diabetes, and that's because a mutation destroyed a gene used by the pancreas where insulin's made. Some people can have lower cholesterol levels and they can't have heart attacks. There's 4 mutations that destroyed the APOC3 gene, its function. So broken genes, beneficial trait. Are y'all getting the point on all of this? Yeah. Some of you guys have new dogs, right? They're coming out with new breeds of dogs. Do y'all like all these new breeds of dogs? They're kind of fun to look at. But the people that have these new breeds of dogs, they have to be at the vet a lot, because the only way you can get a new trait in a dog is by a broken gene. And so we're getting these dogs that's got a whole lot of broken genes, so they have to go to the vet a lot to keep them alive. But we're getting lots of different traits of dogs. So is that evolution? Kinda, but it's broken genes. We're not adding on any DNA. So I just gone through several examples there. Size, 6 different genes, 3 involved with degrading changes in the protein coding region of the gene. The other 3 have tweaks in the control region that lowers the amount of protein. My wife has a genetic disorder. She has beta thalassemia, and her red blood cells aren't made right. You have 2 alpha and 2 beta proteins that make up your red blood cell. Well, her betas are not made right. Well, she's got the gene, but she doesn't have the control region for the gene. And so a break can occur in the gene or in the control region of the gene, or maybe in another protein that causes that one to work. Maybe it's got a break in it or something. But that's how different traits come to be, is because of genes breaking. My whole point in all this is, now that we can do the genetics, every example evolution uses, it's always a broken gene. And so to go from bacteria to what we have today, bacteria has 1,000 genes. You and I, we have 60,000 genes. There had to be addition of DNA along the way, but that's not what happens. It's not what happens. Evolution can only show broken genes, and that's really, really fun. Here's my conclusion. Almost all beneficial mutations are broken genes. The more genes that are degraded for short-term evolution adaptation, the fewer that are available for future adaptation. Once that polar bear had these broken genes, it's pretty much limited to that area. Can't take any more. Natural selection only works at the genus species level. No evidence for the creation of new genetic material. No evidence for the evolution of simple forms of life to complex forms of life. Again, God made all the DNA at the very beginning. He made every organism. We just had a few genes break and it causes new species, but they don't go very far from there. Does that make sense to everybody? Okay, so is there evidence for creation? Yes, there is. Look at this. You'll love this. Then we'll be through. Y'all know Hebrews 11 says this: by faith, we understand that the worlds were prepared by the word of God, so that what is seen was not made out of things which are visible. All God had to do was just say the words, and it happened. It says the same thing in Psalms 148. You and I sing this song all the time. Hallelujah, praise Jehovah. Y'all know that song? Second verse. It's in verse 5. For he commanded, and they were created. All God had to do was just say it, and they were created. Old Testament, New Testament, both confirm that Genesis 1 is true. John 1, in the beginning was the Word was with God, the Word was God, he was in the beginning with God. All things were made through him, and without him was not anything made that was made. In him was life, and the life was the light of men. So everything in the Bible confirms God as our creator. So can we see that power? Yes, we can. These are plant hoppers. This is a plant hopper. There he is. And look what he's got on his back legs. He's got gears. They fit together just like gears in a machine shop. Is that not crazy? Yeah. So that's showing the gears on a nymph, a small plant hopper that hadn't gone to the adult stage yet. But what happens is, to jump, he cocks his back legs and those gears are staying together. Then when he jumps, he can jump really fast, but the gears help both legs jump at the same speed so that he can go right where he wants to jump. Isn't that cool? Yeah, it's really funny. The National Geographic writer in their article said, this insect has gears. He's surprised because where do you see gears? In motors, in machine shops. Yeah, that's where you find gears. God made gears. He made this insect with gears. And so how did that come? And they're even, you know how gears are kind of bowed a little bit so that they fit together when they're turning together a bit? That's the way these are as well. So it's just really, really fun. The eye. I love the eye. I love looking at the eye. This is an article from the Proceedings of the National Academy of Sciences. You'll notice my note down there at the bottom. The eye has always been amazing in its operation, and y'all know that. It is equally amazing to me how science views the eye. They say it is imperfect because of the blind spot and because the photoreceptors at the very back of the retina. Both are dubbed illogical, and a creator wouldn't do that. It's really interesting today in our textbooks. They're even saying now in our textbooks, a creator would not have done this. That's what they're saying in our textbooks. So they're attacking God in our textbooks right now, and that's kind of interesting. But here's the deal. Y'all can see the eye there. So light comes in on the eye here. I knew I was going to do that. Light comes in, and it strikes the retina back here. And that's where the image is at. Now we got a blind spot. So there's a spot in there that we can't see anything. And here's the retina. Oh, there's the retina. And y'all can see the cone and rod cells are here at the back of the retina. And science says they should be up here so that they can get maximum absorption of light. So they say that's kind of imperfect. And for you and I, we'd say, yeah, that kind of makes sense. Well, number one, is anybody bothered by their blind spot? Can y'all tell me where your blind spot at? Is it right here when you're looking, or is it right? Can any of y'all see your blind spot? No, none of us can see our blind spot. I mean, if you put a dot on a piece of paper and move it like this, it'll disappear. Your blind spot will show up somewhere over here. But that doesn't affect us. But the rod and cone cells, it does make sense they should be closer to where the light hits it rather than way out here and all this tissue going through. Well, they found these Mueller cells in the eye. The Mueller cells, they act as an optical fiber, and when the light strikes the retina, it actually, these cells take the light and divide it up to the proper rod and cone cell. So it'll take the red-green lights to the cone cells. It'll take the blue-purple lights to the rod cells. It separates the light like it's supposed to be separated so that we can get maximum absorption of that light. So actuality, we're designed to have maximum absorption by having these Mueller cells take the light and direct it to the right rod and cone cell. Is that not cool? So it's our eyes actually are seeing the best that they can see. That's just another picture from Marcus Eberlin's book Foresight, which I've got back there at the back. He's really good. I'll talk to him about him later. Each cone cell has its own dedicated fiber cell, and these cells actually improve daytime vision, and it doesn't hurt nighttime vision at all. So it seemed to be a design feature, which brings up the point. Anytime you see a purposeful arrangement of parts, that indicates design. Like the clotting of blood. When you have a purposeful arrangement of all these proteins that work against each other to finally clot the blood, that indicates design. When you have a motor on a flagella and all these proteins are working together to make that thing spin, that's a purposeful arrangement of parts. That's design. So isn't that interesting? A few years ago, Dr. Tim Dallas from Tech, he's an engineer over at Tech. I saw in the paper he was going to be talking at a church on the scientific and engineering evidence for creation. And I thought, this is a tech engineer? I got to go hear him. So I did. I went and heard him talk, and he talked about the peacock feather. Y'all like peacock feathers, right? What he called it was bio-nanophotonics. Dr. Dallas works in tiny, tiny little structures. He built a clock. And it had the guns up hands on it. And the clock was only 2 millimeters big. And it was a working clock. So he works in little bitty stuff. And so he calls this bio-nanophotonics on the peacock feather. That's what you and my eyes see, but there's no pigments in a peacock feather. None. There's no pigments. Why do we see different colors to make an eye? When you get down to the microscope level, that's all you see is these photonic lattices is what they call it. They're just photonic lattices is all they are. And here's what happens. When you have a photonic lattice, it may look like that. It absorbs all the colors, but lets one color go through. If you change the lattice, I'm going to change it, it lets another color come through. So that whole peacock feather is different arrangement of lattices to give it the eye of the peacock that we see. This was investigated not by Dr. Dallas at Tech, but by some Chinese researchers in the Proceedings of the National Academy of Sciences. And look what they say. You can see it in purple there at the bottom. They said coloration strategies in the peacock feathers are very ingenious. That's an intelligent design term. They're actually saying this was developed, this was designed. This is just a design feature. Is that not cool? It's just really cool. So that's really kind of fun. This is the Madagascar orchid. So that's an unusual orchid. It's very pretty, but it's got this really long spur. Y'all can see it sticking down there. A long spur that's about 11 to 16 inches long. And at the bottom of that spur is a really tasty nectar for a moth. Well, what moth is going to be able to get that at the way down at the bottom of that spur? This guy. It's the hawk moth. And look at his proboscis. It's 16 inches long. And so he goes to the flower. You can see there how long his proboscis is. He sticks it in the flower and sticks it all the way in and goes all the way down and gets his nectar. You can see there on the left, he's got his face sticking in the flower, getting that nectar at the bottom of that spur. But at the same time, he's getting pollen dust all over his face. So he'll pull out of that one. You can see him pulling out of that flower. He'll go to another flower, stick his proboscis down in there, and the pollen on his face pollinates that flower. That's the only way this flower is pollinated. Is that not cool? How do you get a plant have this design and an animal have this design that they're working together to make something work? That has to be design. That has to be a designer. To have random mutations in two different species, animal and plant, to come together like that? No, that doesn't happen just by random mutations in natural selection. That has to be designed. Here's a manuka flower. And I'm not sure where this is at. I want to say this is in Madagascar. I'm not really sure. But you can see the flowers there. And these flowers, there's a whole bunch of them on that picture. The ones at the bottom you can see have opened up. So here's what happens. This flower, when the pod gets mature, it's got two little petals at the top of the flower. And y'all can kind of see them. They're more the lighter colored, not the green color, but the lighter colored. And these petals kind of open up like this and form a dish. A bat echolocates off that dish, and in between the two petals is a slit. The bat will come and stick his tongue in that slit. And when he sticks his tongue in that slit, that triggers the flower to open. But he's traveling at such a speed, he hits that, and it swings his rear end around. Let me show you. There you go. It swings his rear end around, and when that flower is triggered to open, it dusts pollen on his rear end. And then he'll go to another flower, hit it, swing around, the flower parts all fall down, and the pollen on his rear end pollinates that flower. Isn't that cool? He hits, what is he? Yeah. So there you can, he's just the right size to fit right where those flower parts come down in order to carry pollen from one to another. It takes 0.2 seconds per flower. He hits 100 flowers per night. That's the only way this particular flower is pollinated. Is that not cool? Again, you get purposeful arrangement of parts. That's design. This is my last one, and we're done. Oh, I'm going to be done by 2 o'clock. That's great. This is the hammer orchid. This is one of my favorite. It is an orchid down in Australia. It's got a flower on it. So that is the flower, but it's also got this structure on it. Now that structure is kind of interesting. Here in Australia, you also have a wasp. There is the wasp, but you only see the male thynid wasp. You never see the female. That's because she lives underground. One time a year, she will come out of the ground, she'll walk up a plant, she'll release a pheromone to attract the male. The male will come and mate with her, and she'll go back down in the ground, lay her eggs, and you won't see her till the next year. So the only thing you ever see down there is male thynid wasp. Well, this orchid, it's so fun. This orchid, at the very same time, its flower is about to open. The orchid releases the same pheromone. The orchid has, I'll go back, has this part on it that looks like the female thynid wasp. He will try to mate with that, we call it a dummy female in the plant world, that dummy female, and that is a hinge. When he tries to mate, it'll bring him around. Isn't that fun? It brings him around and dusts pollen on the back of his abdomen. When he figures out this is not working, he'll leave. Another plant will release the same pheromone. He'll try to meet with that dummy female. It'll bring him around, and it pollinates that flower. It's the only way this flower is pollinated. Now, you tell me that came about by random mutations over time. A wasp and an orchid having the same parts, and that orchid has to have the arms of that thing have to be the right distance apart, and that hinge had to have the right torque to it, to that when the male tried to mate, that it would swing him around. You'll see how many parts are involved here? That has to be designed. That has to be designed. And I think that's why God is always saying, just look at what I made. You can tell this had to be designed. We see unique mechanisms of sexual reproduction. We see purposeful arrangements of parts. We see irreducibly complex systems. All these indicates design. That's why God said this. For what can be known about God is plain to them, because God has shown it to them. Ever since the creation of the world, his eternal power and divine nature, invisible though they are, they have been understood and seen through the things he has made. We just need to take a closer look at what he's made. And I think when we take closer looks and we see some of these things that science is telling us, it just shouts there had to be a creator out there. Okay, for you guys I was talking with at lunch, I follow to get all of my information. I've learned over the years who to follow and who not to follow. In the intelligent design world, I follow evolutionnews.org. I know that sounds like an evolution site, but it's not. It's an intelligent design site. And they're really good. They're really good. I will warn you, they do not deal with the age of the earth. They just don't even mess with that. But they look at design. They look at things that are designed, and they show there had to be a creator. And so they're really good at that. It's actually the discovery.org group that is running that, but the website where they keep up with all information, if you see something about evolution, if you go to evolutionnews.org, they will tell you about it from a creation type point of view. Does that make sense? In the church, David Reeves, and for you guys at lunch, it's davidreevesministry.org. So just go there. He's got a museum up close to Nashville, and it's just wonderful. He's got a channel on the TV, some TVs, I think Roku TVs or something. He's got a channel that continually runs different programs, and they're all really good. They're all really, really good. And so he's a good resource to have. Apologetics Press, that's in the church as well. And all those guys do a good job. And so I like their stuff as well on all that. So that's some sources for you that you buy some stuff for your grandkids or something. Yeah. Does that make sense? Yeah. Well, this has been fun. Thank y'all. This has just been really, really fun. I hope you got a lot out of it today. And I'm looking forward to tomorrow. We'll see y'all tomorrow as well. Yeah, I'll turn it back to you guys.
Again, thank y'all for being here. Dr. Martin, thank you.